Where are memories stored? You might have just said, in your head, your brain, your mind? Well, what if I told you that memories can be stored without a brain? Or better yet, what if I told you that memories could be stored after regrowing a brain that's about the size of a grain of sand? Well, that's where planaria worms come into play. Planaria worms are tiny flatworms with the extraordinary ability to regenerate from just 1/279th of their original body. They can do this mainly because of the presence of neoblasts, or planarian stem cells that can differentiate into different kinds of specialized cells quite quickly. Currently, our understanding of human memory surrounds synapses, or the connection between neurons in the brain. As new connections are made, and when those links are strengthened, new memories are cemented and reinforced. In 2013, a study was conducted at Tufts University in order to see if these planaria worms could remember without a brain. Here's how it was done. The planaria worms were split into two groups, with one fed liver consistently on rough-bottom petri dishes and the other on smooth-bottom dishes, creating a food environment memory. After 10 days, all worms were decapitated, allowed to regenerate, and tested for memory recall. To do so, a piece of liver was placed on a quadrant of the rough-bottom petri dish under bright light, and one worm was added to the dish. The time taken for the worm to initiate feeding was recorded, and this was repeated for all of the worms. The worms that had been fed on the rough-bottom dishes before decapitation fed significantly faster than the control group, and so this demonstrated memory retention. And so, last year, I took to a little investigating myself. What percentage of a planaria's body could be regrown and still retain memories? Half of the worms were cut below the auricles, and so 10% of the body was regrown. The other half were cut below the pharynx, and so 60% of the body was regrown. And from there, they were given time in order to recuperate, and they were tested to see how fast it took for them to initiate feeding. Let's take a look at the data. For each percentage of body tissue regenerated, the average control and familiarized time to initiate feeding was put through an ANOVA statistical analysis to test if there was a significant difference between the two values. Since both p-values were less than 0.05, this difference was statistically significant and displayed that the difference was not likely to be caused by chance. So, not only can planaria remember without a brain, they can remember after regrowing 60% of their body. How is this done? The first theory is that memories are held in the peripheral nervous system, which is the part of the nervous system that lies outside of the brain and the spinal cord. And so here, the idea is that the food and surface memories were not held in the brain, but instead they were held in the peripheral nervous system. And so when the body and the brain were regrown, the memories were still intact. This theory really highlights the possibility of untapped capabilities of the peripheral nervous system, which is very exciting. In the second theory, the brain is regrown as a tabula rasa, or as a blank slate. In this case, the peripheral nervous system still holds the memories, but this time, it restores it into the brain, kind of like when a CD is burned with information. Sometimes, the big answers can be found in the little things.